This is a short tutorial to learn how to tether a transcription factor to a piece of dynamic DNA. So to do this, we're going to use mMaya and the double-stranded DNA kit, both of which you can find on clarify.com. And a great place to start is any structure that is a co-crystal of DNA and your transcription factor of interest. So in this case, I've already brought in the 1 NKP transcription factor PDB, which is a co-crystal of MYC and MAX, these two proteins that are binding as a dimer to a strand of DNA at the bottom. And I've rendered a little sequence of the kind of thing we're going to generate. So I just wanted to show you what this looks like. So the steps involved in doing this are pretty straightforward. The basic workflow before we jump in is number one, to pick your molecular representation for the transcription factor. So we'll do that in mMaya. Then we're going to synthesize a dynamic piece of DNA based on the helical axis that we're going to extract from the PDB's DNA. And we might adjust the helical axis a little bit also to make sure that the grooves align, right? And then we're going to customize, if we want, the representation of the DNA. And then finally, to make sure that the transcription factor is properly bound and moving along with the simulated piece of DNA, we're going to create a strand mount and then create a parent constraint between the transcription factor and the DNA. So let's jump in. I'm going to select my proteins in this case and use a secondary structure representation. So I'll ask mMaya to build that representation for me. And just for ease of working, I'm also going to extract that. You can see it creates a new group in the outliner with just the geometry. So I'm working with a piece that won't still be connected to the PDB import kit. It's a little bit easier that way. So that was step one. The next thing I'm going to do is extend this piece of DNA. So again, what we're seeing right now is the chains of the DNA that are from the PDB that I imported. But what I'm going to do is go to my double-stranded DNA kit and go to the tab from PDB. So with the two DNA chains selected, I'm going to say extract helical axis. You can't see it under there, but actually you can barely see it. There's a curve that's been generated, and that curve is now the same length as the imported piece of DNA. So the whole point is to extend it from this. So I'm going to go to my curves menu set and extend that. And sometimes, depending on the piece that you're extending, you might have to readjust this piece if you want a more, like a linear piece of DNA. So I might actually raise that up and do something like this with it. It looks like right at the bottom of this curve that there are some points that were angled downwards because of the shape of this nucleotide right here. So that's not a problem. I can just edit that a little bit and see what we get. Okay, so it's a little bit bent still. I'm going to grab another CV perhaps and go like this. So now that I've done this, let's see, if I select the curve again and press that same command, it'll extend it even more. So that looks like a decent piece of DNA to work with. So with this curve selected and still in my DNA kit, if I go to the Synthesize tab and Build from Selected Curves, I click Build. So now I have a strand that's been created on this new curve. The only issue now, before I hide the PDB based DNA, is I want to make sure that the helical axis of this new dynamic curve matches. In other words, that the grooving matches. So for that, I can do kind of a command middle mouse button over the helical axis rotation offset, and let's see what we get. See, so I know that this basic region here of the protein is binding in the DNA's major groove. And right now it looks like our dynamic DNA, if you see right here, this is the minor groove. So I'm going to rotate that a little bit and make sure that my grooves are lining up. And as I've said before, depending on what you're doing, you may want more or less precision on this. I mean, I'm just matching them by eye just to make sure that what I'm really concerned about is that this alpha helix does not bump into the DNA but sits there in the major groove on both sides. Remember, this is a dimer. So that looks pretty good. And now that I've done that, I can basically go ahead and hide my originally imported PDB. I'll just go to asset visibility and turn that off. So what you're seeing now is the newly synthesized piece of DNA, the secondary structure that I'm going to smooth that, press 3, that we built from the PDB, and we're ready to go to the next step. In this case, I'm not going to play too much with the representation of the DNA. Of course, you could do that in the dsDNA kit by changing things like representation or color sets, but I'm not going to take the time to do that today. So by default, the DNA that we synthesized is dynamic, meaning that if I just press play, it's going to start to simulate. And the goal is, how do we get this transcription factor to ride along with the dynamic strand of DNA? So that's our last step, and it's pretty straightforward. With the DNA selected in my Edit dsDNA tab, I can go to Strand Mounts and add a strand mount. It's going to ask me if I want to, lock the base pair count, and I'll say Lock Now. And with this strand mount created, it will usually create it at one end. I can use the base pair position to move it along the strand. So I'm going to zoom in a little bit and just slide it until it's roughly beneath or as close as possible to the position of my protein. And so with the strand mount selected, I'm just going to shift select the proteins, and now I'm just going to create a parent constraint. And that should be enough to basically now play back. And so what happened there is that in the way that I selected my proteins to do the parenting, I should have selected a node in the outliner. So let's do that again. I'm going to start by selecting the strand mount, and then I'm going to shift select this group. Actually, it's this group up here, sorry. So select the strand mount, command select the ribbons, and then go to create my parent constraint. And now, if I play back, you get the behavior that you're expecting. And this is the same render as I had shown here. So that's it. It's a pretty basic process that's made relatively straightforward with the double-stranded DNA kit.